Hello everyone, my name is Kapil Raikwar and my topic for presentation is structure and function of antibodies. In this presentation, we will be discussing on what are antibodies, structure of antibodies, function of antibodies, classification of immunoglobulin and summary. So let's begin with our first topic, what are antibodies? The antibodies are specialized glycoproteins that are found in response to an antigen and react especially with that antigen. Antibodies are also known as immunoglobulin and are mainly found in serum, vascular fluids, exocrine secretion, and on the surface of lymphocytes. Each antibody are produced by specific B lymphocytes. So let's discuss the structure of antibodies. The structure was given by Porter and Aldman in 1959. Antibody or immunoglobulin molecules are glycoproteins composed of one or more units, each containing 4 polypeptide chains. It has two identical light chains of 25 kilodalton each and two identical heavy chains of 50 to 73 kilodalton each. One immunoglobulin contains one type of light chain and one type of heavy chain. Heavy and light chain are held together by a combination of non-covalent interaction and covalent interchange disulfide bonds, forming a bilaterally symmetrical structure. The amino terminal end of the polypeptide chain show considerable variation in amino acid composition and are referred to as variable region to distinguish them from relatively constant region. Variable region extend from N-terminal and amino acid sequence in this region is highly reliable, whereas constant region extend from end of variable region to C-terminal and amino acid sequence is relatively constant. A chain of arms extend into Hing region which is rich in proline and cysteine. The Hing region is the area of the H chain between the first and the second C region domains and is held together by disulfide bonds. This flexible Hing is found in immunoglobulin G and immunoglobulin D, which allows the distance between the two antigen binding sites to vary. Now, the diagram that clearly shows where the light chain and heavy chains and the Hing region are labeled. Now move to the function of antibodies. Antibodies are secreted into the blood and mucosa where they bind to an inactive foreign substance, pathogen and toxin. Antibody activate the complement system to destroy bacterial cell by lysis, that is punching holes in the cell wall. Antibody facilitate phagocytosis of foreign substances by phagocytic cells. This diagram shows how neutralization and opsonization take place. Antibodies bind to and inactivate viruses and toxins. These antibodies are called neutralizing antibodies. When antigen antibody complexes are activated, they activate the complement system, which triggers its antibacterial activity. In opsonization, phagocytic cells grab the antibody bound to the surface of foreign substances for efficient phagocytosis. Now let's discuss the types of immunoglobulin. Based on the structure and antigenic nature of H chain, the immunoglobulin are divided into 5 classes. The first is immunoglobulin A, which have alpha chain. Immunoglobulin D have delta chain. Immunoglobulin E having epsilon chain. Immunoglobulin G having gamma chain. Immunoglobulin M having mu chain. All of them are similar in structure and are connected by disulfide bonds, but basic structure of immunoglobulin M and immunoglobulin A is quietly different from the other three. Immunoglobulin M is a pentamer, whereas immunoglobulin A is a dimer, where rest of the immunoglobulin are monomers. So let's discuss them one by one with their functions. Immunoglobulin A, it is a dimer which constitutes 10 to 15% of the total immunoglobulin. It is present in milk, saliva, tears, mucous of the respiratory tract, digestive tract, and genitourinary tract. It has a half-life of approximately of 5 days. In serum, it acts as a dimer. In external secretion, it acts as a dimer called secretory immunoglobulin. Functions are: it provides local immunity, it provides protection to the newborn, it binds to the viral and bacterial surface antigen, preventing adherence of antigen. Now we come to immunoglobulin D. It's a monomer which has two epitope binding sites which make up approximately 0.2% of the serum antibodies. It is mainly found on the surface of B lymphocytes. Function of immunoglobulin D are: it helps in B cell activation and also act as a receptor for antigen binding. Now we come to immunoglobulin E, which have epsilon chain, which found in low concentration and is present in serum and has half-life of 2 to 3 days. A function of immunoglobulin ERS. It is responsible for immediate hypersensitivity. It binds to Fc receptor in basophils and mast cell. Release of substance like histamine as vasoactive mediator. Now we come to immunoglobulin G. 80% of the serum antibodies are immunoglobulin G. Half-life is of 12 to 23 days. It's a monomer and has two epitope binding sites. It has further four subclasses which are as immunoglobulin G1, immunoglobulin G2, immunoglobulin G3 and immunoglobulin G4. Function of the immunoglobulin GRS: it neutralizes the poison on the toxin. Its immune response appears late and persists for longer time. It is bacteriolytic, versatile and precipitating antibody. This type of antibodies can readily cross the placenta and play an important role in protecting the development fetus. Now the last one, immunoglobulin M. It is the largest antibody which accounts for 5 to 10% of the total serum proteins. It is mainly found on the surface of B cell and has half-life of 5 to 6 days. It has 10 total antigen binding sites. It is a pentamer which is a polymer composed of 5 monomers. Function of immunoglobulin M are: first, it activates complement of classical pathway. It provides defense against multivalent antigen. It acts as opsonin. It agglutinates bacteria. So, as we have come to the end of the topic, so we will summarize what we have learned. Antibodies or immunoglobulin are the molecule secreted from plasma cells that mediate the humoral immune response. There are 5 antibody classes. An antibody class determines mechanism of action and production site but does not control its binding capacity. Antibody bind antigen by variable domains and can either neutralize pathogen or mark them for phagocytosis or activate the complement cascade. This was all about structure and function of antibodies. Thank you.